For centuries, we've assumed the human body operates exactly like a machine. You use it, the parts experience friction, they wear down, and eventually they break. But your biology tells a different story. If you look at the genetic code of an 80-year-old, the DNA molecule itself remains virtually identical to the DNA they had as a teenager. The parts haven't actually worn away at all. Aging is a cellular identity crisis. To understand why, we have to look at the body's software, known as the epigenome. Think of the epigenome as a set of chemical tags attached to your DNA. These tags act as instructions for a blank cell, telling it whether it should become a skin cell, a nerve cell, or a liver cell. The problem is our daily environment. Every time you get an X-ray, eat highly processed foods, or even lose a single night of sleep, you create microscopic damage inside your body. Over decades, this constant stress physically knocks those epigenetic tags out of their proper place. The DNA remains perfectly intact, but the operating instructions become chaotic. Aging occurs when cells lose biological information and literally forget what they're supposed to do. Harvard professor Dr. David Sinclair has spent the last 30 years mapping exactly how this information is lost, and more importantly, how to get it back. This specific mechanism of aging is actively being targeted and tested at some of the most elite research centers in the world. Dr. Sinclair's research isolates this exact biological flaw, reframing aging as a quantifiable engineering problem. The epigenome's chemical tags don't manage themselves. They are organized by a specific group of proteins called sirtuins, which act like conductors telling the cellular orchestra exactly what to play. But sirtuins have a secondary job. When a DNA chromosome breaks, these conductors must drop everything, abandon the epigenome, and rush over to repair it. Every day, your body experiences trillions of these microscopic DNA breaks. The sirtuins constantly scramble to fix the damage, and in the chaos, many get lost. They fail to return to their original posts, leaving the cell's identity unprotected. This chart shows the fuel problem accelerating the crisis. It tracks a molecule called NAD+, which sirtuins require to function. NAD+ levels plummet by 50% between ages 20 and 50, taking the cell's repairability down with it. Without enough fuel and with constant daily emergencies, the conductors permanently abandon the orchestra. The resulting cellular chaos is what we experience as gray hair, wrinkles, and physical frailty. Our modern lifestyle of predictable meals, air conditioning, and sitting at desks is a stark contrast to the brutal environments our bodies evolved in. Our ancient ancestors survived strictly because extreme conditions naturally triggered deep, powerful cellular repair mechanisms. We can manually activate those same mechanisms today through a process called hormesis. This simply means intentionally reintroducing biological adversity into our lives to trick the body into entering a state of survival. The triggers are practical: skipping meals to induce fasted states, exercising until you are breathless, exposing yourself to extreme heat and cold in an ice bath. This calculated stress forces the body to spike its production of NAD+. With a sudden flood of fuel, the sirtuins return to their posts and resume protecting the cell's identity. Leaning into temporary, controlled physical discomfort forces your biology to fight back. It is the most effective way you can protect your genetic software and slow down your aging clock today. But slowing down the loss of information is no longer the limit. Researchers have discovered that every old cell in your body retains a pristine, hidden backup copy of its youthful software. To access it, scientists have engineered a specialized microscopic delivery vehicle packed with 3 specific reset genes. In a recent laboratory trial, researchers injected this delivery package directly into the optic nerves of completely blind animals. The reset genes successfully accessed the backup copy inside the damaged cells. They turned the biological clocks of those specific cells back by 75% and entirely restored the animal's sight. This age reversal technology is crossing the threshold from animal models into official, government-approved human trials, beginning with patients suffering from severe vision loss. Reversing the age of brain tissue restores the cell's youthful ability to heal itself and clear the proteins that cause dementia. A rejuvenated immune system actively identifies and violently kills hidden cancer cells before they can even take hold. By treating the root cause, the process of aging itself, we neutralize the dark environment these diseases need to exist.